Aneuploidy is a condition in which the number of chromosomes in the nucleus of a cell is not an exact multiple of the monoploid number of a particular species. An extra or missing chromosome is a common cause of genetic disorders including human birth defects. Some cancer cells also have abnormal numbers of chromosomes. Aneuploidy originates during cell division when the chromosomes do not separate properly between the two cells. This generally happens when cytokinesis begins while karyokinesis is still underway. Different species normally have different numbers of chromosomes from one another, and the term aneuploidy refers to the chromosome number being different from the usual number for that species. Chromosome abnormalities occur in 1 of 160 live human births. Apart from sex chromosome disorders, most cases of aneuploidy result in termination of the developing fetus. The most common extra autosomal chromosomes among live births are 21, 18 and 13. Chromosomes: every cell in the human body, apart from enucleated red blood cells and the haploid gametes, has 23 pairs of chromosomes. One copy of each pair is inherited from the mother and the other copy is inherited from the father. The first 22 pairs of chromosomes are numbered from 1 to 22, and are arranged from largest to smallest in a karyotype. The 23rd pair of chromosomes are the sex chromosomes. Normal females have 2 X chromosomes, while normal males have 1 X chromosome and 1 Y chromosome. During meiosis, when germ cells divide to create sperm and egg, each half should have the same number of chromosomes. But sometimes, the whole pair of chromosomes will end up in one gamete, and the other gamete will not get that chromosome at all. Most embryos cannot survive with a missing or extra autosome and are spontaneously aborted. The most frequent aneuploidy in humans is trisomy 16, although fetuses affected with the full version of this chromosome abnormality do not survive to term. The most common aneuploidy that infants can survive with is trisomy 21, which is found in Down syndrome, affecting 1 in 800 births. Trisomy 18 affects 1 in 6,000 births. And trisomy 13 affects 1 in 10,000 births. 10% of infants with trisomy 18 or 13 reach 1 year of age. Changes in chromosome number may not necessarily be present in all cells in an individual. When aneuploidy is detected in a fraction of cells in an individual, it is called chromosomal mosaicism. In general, individuals who are mosaic for a chromosomal aneuploidy tend to have a less severe form of the syndrome compared to those with full trisomy. For many of the autosomal trisomies, only mosaic cases survive to term. However, mitotic aneuploidy may be more common than previously recognized in somatic tissues, and aneuploidy is a characteristic of many types of tumor originates. Terminology: in the strict sense, a chromosome complement having a number of chromosomes other than 46 is considered heteroploid, while an exact multiple of the haploid chromosome complement is considered euploid. Mechanisms: non-disjunction usually occurs as the result of a weakened mitotic checkpoint, as these checkpoints tend to arrest or delay cell division until all components of the cell are ready to enter the next phase. If a checkpoint is weakened, the cell may fail to notice that a chromosome pair is not lined up on the mitotic plate, for example. In such a case, most chromosomes would separate normally, while others could fail to separate at all. This would generate a daughter cell lacking a copy and a daughter cell with an extra copy. Completely inactive mitotic checkpoints may cause non-disjunction at multiple chromosomes, possibly all. Such a scenario could result in each daughter cell possessing a disjoint set of genetic material. Meroteric attachment occurs when one kinetochore is attached to both mitotic spindle poles. One daughter cell would have a normal complement of chromosomes. The second would lack one. A third daughter cell may end up with a missing chromosome. Multipolar spindles, more than 2 spindle poles form. Such a mitotic division would result in one daughter cell for each spindle pole. Each cell may possess an unpredictable complement of chromosomes. Monopolar spindle, only a single spindle pole forms. This produces a single daughter cell with its copy number doubled. A tetraploid intermediate may be produced as the end result of the monopolar spindle mechanism. In such a case, the cell has double the copy number of a normal cell, and produces double the number of spindle poles as well. This results in 4 daughter cells with an unpredictable complement of chromosomes, but in the normal copy number. Somatic mosaicism in the nervous system. Mosaicism for aneuploid chromosome content may be part of the constitutional makeup of the mammalian brain. In the normal human brain samples from 6 individuals ranging from 2 to 86 years of age had mosaicism for chromosome 21 aneuploidy. This low-level aneuploidy appears to arise from chromosomal segregation defects during cell division in neuronal precursor cells, and neurons containing such aneuploid chromosome content reportedly integrate into normal circuits. Somatic mosaicism in cancer. Aneuploidy is consistently observed in virtually all cancers. Somatic mosaicism occurs in virtually all cancer cells, including trisomy 12 in chronic lymphocytic leukemia and trisomy 8 in acute myeloid leukemia. However, these forms of mosaic aneuploidy occur through mechanisms distinct from those typically associated with genetic syndromes involving complete or mosaic aneuploidy, such as chromosomal instability. Therefore, the molecular processes that lead to aneuploidy are targets for the development of cancer drugs. Both resveratrol and aspirin have been found in vivo to selectively destroy tetraploid cells that may be precursors of aneuploid cells, and activate AMPK, which may be involved in the process. Alteration of normal mitotic checkpoints are also important tumorigenic events, and these may directly lead to aneuploidy. Loss of tumor suppressor p53 gene often results in genomic instability, which could lead to the aneuploidy genotype. In addition, genetic syndromes in which an individual is predisposed to breakage of chromosomes are frequently associated with increased risk for various types of cancer, thus highlighting the role of somatic aneuploidy in carcinogenesis. It has been suggested that aneuploidy might directly contribute to carcinogenesis by disrupting the asymmetric division of adult stem cells, thereby leaving those cells capable of limitless expansion. Partial aneuploidy. The terms partial monosomy and partial trisomy are used to describe an imbalance of genetic material caused by loss or gain of part of a chromosome. In particular, these terms would be used in the situation of an unbalanced translocation, where an individual carries a derivative chromosome formed through the breakage and fusion of 2 different chromosomes. In this situation, the individual would have 3 copies of part of one chromosome and only 1 copy of part of the other chromosome involved in the derivative chromosome. Aneuploidogens. Agents capable of causing aneuploidy are called aneuploidogens. Many mutagenic carcinogens are aneuploidogens. X-rays, for example, may cause aneuploidy by fragmenting the chromosome. It may also target the spindle apparatus. Other chemicals such as colchicine can also produce aneuploidy by affecting microtubule polymerization. Diagnosis. Germline aneuploidy is typically detected through karyotyping, a process in which a sample of cells is fixed and stained to create the typical light and dark chromosomal banding pattern, and a picture of the chromosomes is analyzed. Other techniques include fluorescence in situ hybridization, quantitative PCR of short tandem repeats, quantitative fluorescence PCR, quantitative PCR dosage analysis, quantitative mass spectrometry of single nucleotide polymorphisms, and comparative genomic hybridization. These tests can also be performed prenatally to detect aneuploidy in a pregnancy, through either amniocentesis or chorionic villus sampling. Pregnant women of 35 years or older are offered prenatal diagnosis because the chance of chromosomal aneuploidy increases as the mother's age increases. Recent advances have allowed for less invasive testing methods based on the presence of fetal genetic material in maternal blood. Types, see also, chromosome abnormalities, ploidy, polyploidy, references. External links, aneuploidy testing, aneuploidy FAQ, genetics of aneuploids.